Welcome to Fuzzybutts and Friends. Well, as much as it drives Fuzzybutts all over this place, absolutely crazy when they hear me say it. I am the host of the show, your big dog, Luke Robinson, and I'm back again with you this week. And look over to my left, who we have back again with us. It's Ginger Morgan. She's the co-host and co-producer of the show, and she's also the director of the Puppy Up Foundation. Ginger, how are you?
You didn't call me your little dog.
I,
You're just not gonna, you're not gonna get that, are you?
that's a
I'm your
branding
little
issue
dog.
on. I have a hard enough time branding your big dog and all the things that I try to do. So I'm a mess when it comes to promoting myself. You have to promote yourself. It's a lot, as you know. It really is a lot to get a brand or yourself promoted. I think, especially out there on social media. But we talked about this yesterday, Ginger. I'm very excited for our guest today and interested and fascinated. And I'm also hopeful as well, because as you know, 2 years ago, almost 3 years ago, will be this year, I lost Hudson to mast cell cancer. On the upside, he was a long-term survivor of it. I think 10 years is what we talked about. So if you look at it in the broader sense, we got a good medical outcome. But what's frustrating is there's just wasn't any other alternatives other than the traditional ones. So we have a wonderful veterinarian to talk to today, Ginger. It's Dr. Pamela Jones with QBiotics. How are you? Welcome to Fuzzybutts and Friends, Pamela.
Great. Thank you, Luke. I'm looking forward to sitting down with you. Thank you for the invitation.
Yes, it's exciting. I can't wait to get into the science side. I can't wait to talk about the results of your clinical trials. But before we get into all the nerdy stuff that I like to talk about, we like to start off with a little bit of levity. Tell us about your Fuzzybutts. Do you currently have any?
I have two, and one is like Velcro. I laugh at your name, Fuzzybutts, because that's exactly what he is. He's an Australian Shepherd, Quinn. And Quinn, we rescued him when he was about a year old, and he's just the love of our lives, you know. And then the other one is actually a young pup. I say young because she's 11 months, which means she still has all the bad habits. And she's actually a standard Schnauzer,
Wow.
the middle size, which you don't see that often.
What's her name?
Her name is Sydney.
Cindy or Sydney?
Sydney. Yeah, Sydney. Yeah, she keeps us very busy and very much on our toes. And she keeps Quinn on his toes too. For him being, 12 years old now, he is just as spry as ever because of her.
But the Schnauzer keeps the Aussie on his toes. That's kind of
That's
the reverse.
my experiences with Schnauzers, whether they're mini, the standard or the giant, they get into trouble at whatever age they are.
Yes.
I have one that her mother passed away, and I was given her in her mom's will, and she's 11 going on 12. She has some heart issues, but boy, let me tell you, she runs and she still has the zoomies in the backyard like she was 11 months, not 11 years. So good luck with that.
Oh yeah, absolutely. Our last one lived to be 14, and she was a cancer survivor as well for a disease that they get. And I mean, amazing the energy she had at 14. They just keep going and going. Yeah, little athletes.
What was her name and what was the type of cancer?
So Abby was a standard Schnauzer, and she had squamous cell carcinoma of the digit, which is truly of the nail bed of the digit. And she actually, in her lifetime, had 4 toes total amputated. Yeah, pretty phenomenal. I discovered the first one when she was only 8 years old.
Wow.
But I think that's the life of an oncologist, is when you discover it before it's a problem, you take care of it immediately, very low stage. Because I even said to one of my colleagues, I said, all the prognosis stuff is horrible for these. And she goes, okay, let's bring you back to being an oncologist instead of a pet owner. She said, if you catch it early. And I'm like, I know. And 2 years later, she had a second one. It was like, well, it's on another foot. Let's take another digit off. And she was still running half marathon distances.
Wow.
So is squamous cell carcinoma of the toenail common in the Schnauzer breed? Is that correct?
Yeah.
And specifically of the toenail.
Yeah, and it's mostly the standards and the giants. And the other one is they get melanomas of the ungual crest of the nail bed. Yeah.
And what is the thinking about that, or is there any thinking why that breed is afflicted by that specific no theory?
Yeah, it's going to come down to mostly genetics, of course. But we don't, to my knowledge, still we have not identified the exact gene that drives that. And it is interesting because it does arise from that growth, that nail bed.
Yeah, well, we have a hard enough time getting studies for the big one, the top 5 cancers,
Yeah.
let alone squamous cell of the toenails. That's very, I would imagine that's pretty rare, thank goodness. But we mentioned early diagnostics and how important that is. And as before we press record on this podcast, you mentioned Dr. Andy Flory with PetDX. And thank God there's a company out there that's at least promising and delivering results, great promising results. We've taken the Onco Canine Ginger 6 times, I think, with between us with our kids, and I'm not sure if squamous carcinoma is on that list of ones that they test for, but
Yeah.
it will be one day. One day, at one point they'll have all the targets there. So
I
uh,
agree. I agree.
I always,
Luke, honestly, when I started as an oncologist, I wish I had a nickel for every time a client would come in and say, can't you just run a blood test and tell me if my dog has cancer? And I'd be like, well, and they'd be like, well, they do it in humans. And I was like, well, the similarity in humans are things like PSA levels in men, with prostate cancer, and you can follow those levels. There's other ones in women for ovarian cancer and such. But yeah, and I have to explain to them, no, basically we have our hands, our tools of doing a physical exam, our, chest x-rays and our ultrasounds and CT scans. But unless it's a certain size, we're not going to find it. And that's what liquid biopsy allows, is you can find it when it's very small.
Absolutely, absolutely.
Yeah.
Well, let's talk about but before we get to talking about the wonderful work and that QBiotics is working on, I want to know about your story, Pamela. I want to learn about your arc. How did you get from the beginning and to QBiotics and the great work you're doing there?
Yeah, I was the typical kid. By 4 or 5 years old, all I wanted to do was be a veterinarian or a pediatrician. So I followed my heart and became a veterinarian. When I went to vet school, I was going to be a mixed animal practitioner because I did that for 9 years as a technician. 7 years, sorry. And I spent that 7 years just seeing so much variety. I was like, why would you not do anything else, right? And then as you have it, it's kind of like college, you change your major. And so my first year of vet school, the first dog I ever owned by myself away from my family, it was my dog, truly, she got a nasal tumor. And I was flung into oncology as far as, I just jumped in. And, I found oncology to be both friend and foe. And I know that doesn't sound great. But the reason I found it to be a foe is what everybody does. Ugh, the big C word, we don't want to talk about cancer. We don't talk about cancer in us or our pets. But what I found that was fascinating, and I call it our friend, is think about if we could unleash or unharness and figure out how those cells learn to divide again and how they change their genetics. We could harness that to do so many different things in human medicine. And so just fascinating to me. And so that's from then I went into medical oncology, and then I did radiation oncology. And my biggest thing all along was my first and second mentor. They both taught me about clinical trials. And so we were always doing clinical trials. And I found that it brought new and novel products to my patients, so I could always offer more. That was where comparative oncology just, took off in my brain. And since then, I've just kind of followed that path. And that path led me eventually from clinical practice for 20 years to actually working in pharmaceutical industry and now doing exactly what I love. And that is comparative oncology and working for a company that does both human and veterinary pharmaceutical development.
Yes, I was very fascinated when I looked at your pipeline and spent some time doing due diligence on your website that you do both human medicine and veterinary medicine as well. But it's interesting that you wanted to do, or you were interested in pediatrics, but as you know, with comparative oncology, most nearly all of the pediatric cancers dogs get as well. And almost all of the work that's being done on the therapeutic side is being done with the comparative oncology trials on dogs first before we test them on children. So what a great story arc. I find that it's fascinating. This is a great show. We get to meet so many interesting, fascinating people, but I find that the most fascinating veterinarians, especially veterinary oncologists that we interview and talk to on this show, they wanted to be a veterinarian at an early age. It was just that was part of it's almost like it was intrinsic in their DNA. But I do appreciate that you said that there's a friend and foe. I understand that. I love the work that we do with the Puppy Up Foundation, but it's also tremendous. It's hard. It's tough as well.
Yes.
The good news is the uptick is we got so many great, interesting technologies underway on the diagnostics and the therapeutic side. So there's a lot to be hopeful for. So let's get to, so you did clinical practice for 20 years. Where did you do that?
I did part of it in Arizona and then the rest in Texas. So I started out in Arizona for a few years and then migrated my way to Texas, to Austin, Houston, as well as Dallas for a little while.
And so you enjoyed clinical practice, but you just really wanted to be, I guess, upstream in the discovery and actually creating the therapeutics that will be downstream used by patients. Was that?
Yes, and it's amazing because I had so many clients tell me, oh, you're going to miss clinical practice, and the truth is, I thought I was going to be, I thought I'd be kind of miserable missing clinical practice and want to go back, and colleagues of mine have. What I found is, and I tell this to young veterinarians that want to try something different, I'm like, keep your foot in the door. So what I did is my first two companies I worked with, I negotiated to work 2 to 4 days a month in clinical practice. And it was good for them because I still got to use the products we were talking about, right? So I had personal world experience of use. And then as well, it kept my foot in the door and it kind of gave me that taste of things. But I will tell you, once I was about 3-4 years in, I just got that bug to do something bigger. And it really was. It's like, where can I contribute on a larger scale?
And that is on the research and academic side. We have a lot of practicing clinicians out there, but we just need more. We need more on the research side, not only in terms of intellectual assets, but financial assets as well.
We need more in general when it comes to veterinary oncology.
We need bigger buckets for veterinary oncology.
Exactly.
Absolutely. So speaking about that, I almost asked you the question offline earlier, but do you know exactly how many veterinary oncologists there are today? Last time, Ginger, I think we counted it was 525 or something like that. Where are we at today?
Like
how many veterinary oncologists are there in the U.S.?
I don't think we're over 600. And what you also have to realize is that includes some that are retired. That includes some like me that don't work in a clinical practice. So I would guess that probably 400 or more are probably clinical practice. And that's seeing patients every day.
Wow, that's so interesting. I've never discussed it and had anyone round it down that there are some out of that number are taken out of the population, the current population.
Some of those that are members of the Veterinary Cancer Society are not in the United States either. When you go to those conferences, they are from all different countries. So you have to take that part of it. If you're looking at just their membership, you have to take those people out of it as well.
Yeah, because you go to Europe and there's lots of oncologists there, not as many as here. You look at a country like United Kingdom and they probably have 8 to 10 oncologists throughout all of the United Kingdom. So not that much, or not that many. I want to say Italy probably has 6, 7. I think Spain has 4 or 5.
Wow. And the reason why I brought it up, it seems like a non-specific question, but I just like to emphasize to our audience out there, most of which are pet parents, so we do have a lot of veterinarians that listen to the show, is that a lot of them have to travel out of state to a different state to get an appointment with this board-certified veterinary oncologist. But again, I know I do this almost every episode, is please pay the expense, take the trip, the travel, the expense, and the time to go consult with a board-certified veterinary oncologist. It will make a difference in the outcome, I believe, of your Fuzzybutts. So But I know I was singing to the choir, but man, whatever we can do to get more of them. I've talked to Craig Clifford, talked to Phil Bergman, how can we get more? But it's tough. The supply demand curve of that degree of professionalism is a tough nut to crack. All right, let's get on to QBiotics and let's talk about QBiotics first, because I thought you were focused on plant-based small molecule. Science is fascinating. So tell us about the company and how you got involved with them.
Yeah, so the company QBiotics is based in Australia. It's an Australian-owned corporation, and it is a company that literally looks at the rainforest and says, okay, what can we learn from this mega diverse plant forest, and figure out what is happening that is creating the protection for the plants, essentially. And the way I think about it is, and we look at plant animal interactions, we look at plant environment interactions, as well as plant and microbiome interactions, right? And if you think about a plant, just in the simplest terms, if something comes up to eat it, it can't run away, right? So I will just call whatever's eating it is a predator of some sort to the plant, and the plant can't run away. So what does it do? It develops mechanisms to protect itself. And whether that be spines, something mechanical, or something bitter tasting, or something that burns when they chew it, that's how they protect themselves. And ironically, our first drug, Stelfonta, Tigelinol Tiglate, came from the observation that the young critters would pick up the young cassowaries, which is a big bird like an ostrich. They would pick up the fruit of this tree, and they would literally, it's bitter tasting, and they would just, they would eat the fleshy fruit off. They'd get to the nut, and it's bitter tasting, and they'd shake it and toss it on the ground. And so a lot of it is just observing that, wait a minute, they eat the fleshy fruit, but then they spit out the seed, and seeds have tons of, energy source. So why wouldn't any animal eat it? But instead they just simply disperse the seed for the plant.
I think it's fascinating what you talk about the biome and how plants work with the entire surrounding biome. Is that we know, I can't recall the particular species of the ant, but we know that some plants work in conjunction symbiotically with plants.
Yes.
The ants will only eat so much, but they don't eat like the roots, and they keep the plant alive, and then the ants in turn protect the plants from being eaten by other. I don't know if you call them predators, whatever you call them.
Yeah.
Herbivores.
Herbivores. They're herbivores, but like I said, we'll call it a predator for the plant.
Yeah, predator for the plant. I love that. But right there, fundamentally, it speaks that there's a lot chemically going on and biochemically going on in terms of signaling and feedback systems that we just have never detected before. So I find that fascinating that's the basis of your work.
Well, and a very brilliant ecologist said to me one time, okay, stop thinking of it as a plant, and said, okay, think of the roots. What are the roots? I'm like, the roots, it's like the roots, it's a plant. And he's like, no, that's the GI tract of the plant. And I was like, whoa, that's kind of brilliant to think of it that way, because truly that's where they get their water, their nutrients, everything that energizes them comes from that. So the roots are an anchor, but even the microbes that live in that soil are responsible for the health of the plant. So you can really think of it as the GI tract of the plant. And I was just, wow, okay, I see plants completely different now.
And I will for the rest of my life.
Yeah.
But I think you treat it with more respect, though. If you humanize a plant, which we are, because they're complex organic systems, it humanizes them more, and you think, well, I got to feed it. Literally, I got to feed its gullet. But I'm fascinated that you looked at plants in Australia, and how did you find how did you find a compound out of a plant in Australia that became the basis for your work for mast cell tumors in dogs? That's just, you wouldn't normally bring those two ecosystems together, those two universes together.
Yeah. It's really some pretty fascinating bench research. So they take the plant basically, and specifically for this plant, it's the kernel, and they literally extract all the different chemical properties. So they're looking at all the molecules that come out of the kernel. And this is the way a lot of drug discovery happens, is by looking at the different molecules. And then they test them in certain ways to say, okay, what does it do in an in vitro model? So what does it do? Does it cause the cells to die? Does it cause the cells to divide? Does it cause, certain bacteria to die? Does it, so how does it work that way? And when they start seeing it go down that path, they go, okay, this is great. And if you look at tigilinol tigolate alone, there's probably 100 analogs of it. So, and it's not a semi-synthetic, but some of them are semi-synthetic, so they're partially manufactured. And they're better at wound healing than, say, tigilinol tigolate is, but tigilinol tigolate is better at killing tumors than what our wound healing drug would be. And so it really is there's a lot of background science that goes into making an observation in the forest, taking the leaves or the plant or part of the plant in, and figuring out what chemicals are really responsible for what it's doing.
And that's how you discovered the chemical basis, the chemical name for Stelfonta. So before we get into the drug, I guess maybe in the mechanisms of action of it, let's talk about mast cell tumors. Just for not all of our audience are educated, I guess, to the extent that you are, about mast cell tumors and how common they are. So give everybody kind of a basic understanding of mast cell tumors. What are we dealing with?
Yeah, so mast cells are part of the immune response. And what happens in dogs most often is they develop a solid cluster of these bad cells, and they develop a mass on the skin. Now, there's also ones that develop subcutaneously, and sadly, there are ones that actually can be very aggressive. Fortunately, it's rare, but very aggressive and originate like in the bone marrow or elsewhere in the body. And the way I think of it is, mast cell being an immune cells are supposed to be single entities, right? They float around and do their job, right? And what happens is, again, it starts to develop a cluster or a clump, and that develops into a mass. It is the most common cutaneous tumor. So when you talk about skin tumors, it's the most common. And sadly, there are several breeds that are predisposed to them that we see very commonly. And many of those are brachycephalic breeds. I mean, you have your popular ones, the pug, the Boston Terrier, bulldogs, but you also have golden retrievers that fall in that, Labrador retrievers. So it's just one of those. Even Weimaraners, boxers for sure are one. But all of those breeds are very susceptible. And that's not to say I don't see them in other breeds. We do. But if you've got one of those breeds, boy, keep your little red flag up. And anytime you find a lump or mass or anything, get to your vet so they can take a look at it.
Yeah, Ginger often talks about aspirating and the need for don't wait, aspirate, I think is the slogan. It's absolutely important. So let's talk about Stelfonta. So how does Stelfonta work on the mechanism of the histamine, that reaction?
Yeah, so what it does, and Stelfonta doesn't actually directly, it's not a molecule that directly attaches to something on the tumor cell to inactivate or kill the tumor cell. It actually works through several mechanisms. One is along the needle tracks where we get that, because we inject this in the tumor. This is not something we give IV. It's not something that pet takes orally. It's something that we inject directly into the tumor called intratumoral treatment, which is becoming more and more something researched on the human side. And what it does is it first starts killing the cancer cells that are closest to us. We call that oncosis, and it's just a high concentration of the drug, and it disrupts their endoplasmic reticulum and some of the other little things within the cell, the organelles within the cell. The second thing it does is it does stimulate an innate immune response. So now you get macrophages and neutrophils called in to clean up. So it's telling the immune system, we want you to come in here and take care of this problem. And so they come in, and that leads to more inflammation. The big unlock that we did years ago was figuring out that it activates something called protein kinase C, and that is in the endothelium or the blood vessel linings. And so what that does is it causes them to essentially become leaky. So you get leaky blood vessels. And remember, tumors, what some of your readers don't, or your listeners don't know, is tumors do have lots of vasculature, but it's disorganized, it's chaotic, it's not like good blood vessels. So it's very easy to disrupt. And then what happens is essentially that tumor collapses in necrosis, so it dies, okay? So that's the big things that it does to the tumor. But one of the things I think is most interesting to me about the drug is you've just killed a tumor tissue. That tumor falls away, literally. We don't surgically remove it, it falls off. And left behind is this beautiful wound bed for healing, right? And the wound healing properties of this drug are amazing. I mean, it stimulates something called granulation tissue, which is the first thing that starts healing a wound. It actually causes breakup of the bacterial biofilm that forms in wounds. So there's a lot going on there beyond the tumor just dying and falling off. It's a complicated drug. These molecules are getting more and more complicated.
Yeah, they are. It's so was when you was the discovery part of this compound then as originally as a wound healing drug, and then during that I guess the trials or the trials of that you discovered that it had the would you call it then anti-angiogenesis properties that this drug has? Would you refer to that? You talked about that.
Yeah.
Okay.
Yeah, because it doesn't kill new blood vessel growth. What
Okay.
it does is it destroys the existing ones.
Got it.
And actually it was kind of the opposite. We found the tumor targeting piece to it. And as we did more and more in vitro bench research in the lab, it was like, wait a minute, it's got a lot of other properties. And that's actually how one of the other analogs, which you may or may not have seen on our website, is in wound healing studies now, because that drug, that analog, which is like a sister of tigelinol tigolate, that drug itself is actually a very potent wound healer.
So is it targeted therapy, targeted specifically to the mast cell tumors? Is it?
That's the unique thing about this drug is it's actually what we would call pan tumor effective, because it's not, so many drugs now, we have to find the abnormality on the tumor cell to hit that target, right? So we're being very specific. This one is not specific for any certain tumor type. And and we noticed that early on when we first started looking at tumors, we noticed that, boy, it's effective in a lot of different tumor types. The difference in effectiveness is based on, the dose you give of the drug, the concentration dose, as well as, the tumor itself, the vasculature of the tumor, the amount of immune response that you can get within that tumor and such. And that's why we learned so much about it in the beginning, but we had to narrow down, okay, what would give the best indication for use in animals that it could be widely used and be widely effective.
So do you see this compound then as a platform therapy that you can then apply to other types of cancers? Is that how you're looking at this?
That's a good question. Right now, that would be off-label use, but we are doing work. We have what are called phase 4 trials, which means after the drug is available and it's on the market, we design phase 4 trials for other types of tumors. And yes, we are seeing efficacy in other types of tumors. One of the interesting, we talk a lot about comparative oncology. And this is a situation where the drug itself that is marketed for dogs, we have a different concentration that is in clinical trials, human clinical trials right now for 2 other types of tumors.
What type of tumors are those, if you can say?
Yeah, I can say, because it's actually, you could probably even see it on our website, but we're working with Memorial Sloan Kettering on a soft tissue sarcoma trial in humans. And that trial actually is almost fully enrolled, and we'll move on to another, additional trials. And then the other one we're working on is just head and neck tumors. So it's everything from squamous cell carcinomas, melanomas, angiosarcomas. There's a lot of different ones that occur in the face and head of people. And again, those are in what are called phase 2 trials. So we've got the safety and efficacy, and now we're working into other types of tumors for to move on to phase 3 trials. Now, and the other interesting thing is you talk about the soft tissue sarcoma, the FDA, there's something called an orphan drug designation with the FDA. And basically it's for tumors that there's not that many of them. There's really not that many effective treatments. I kind of laugh when they say orphan drug designation. I'm like, oh, that's pretty much where we live in vet medicine, but in human medicine, isn't it?
Absolutely.
But the FDA, we put in an application for that drug. Again, it's a different formulation, same chemical, but different formulation for people. We put in an orphan drug designation for soft tissue sarcomas and got it approved. So it's, we're just finding that kind of efficacious. And the important point here for your listeners is a lot of that is based on what we learned in vet medicine. In fact, that's a huge chunk of the packet is showing them, look, this drug went through clinical trials for mast cell disease in dogs. We got this information from it. We know this information, and it's a successfully marketed drug now.
Are you talking about now that you can take that to the human medicine side? Is that what you're referring to?
That's exactly what we did in the application for orphan drug designation. And orphan drug designation, basically it's a stamp to say, yep, your drug has shown enough safety and effectiveness that you can go it's almost like what I would call a fast track to get approved in human medicine. And literally the biggest chunk of our application was based on what we'd learned in dogs.
It's amazing that you still have
exciting.
It is very exciting, but it's amazing to me, and I'm sure it is to you, that we still have that veterinarians have to educate the human doctors, the human side, medicine side, about the power and the importance of comparative oncology and more importantly, one cancer. It's almost like we really don't think about different species or multi-species cancer. It's one cancer in a shared ecosystem, and there's a basis for doing research on both. It's interesting that Ginger and I went to some years ago. It was a symposium that brought the human medicine, a human oncologist, and pharma and the veterinary medicine together. And this was the very topic, and we were fascinated that there were so many human doctors, but that still didn't know, hey, we should be doing parallel studies, right? Comparative.
Absolutely.
Yeah, absolutely.
I was shocked. I mean, I was just shocked about the whole thing. When the human doctors were like, I didn't realize that dogs got the same type of cancers, and then you use the same x-rays and the MRIs and all that stuff, the same machines that we use. I was just like, how do you, I felt like I'm like this lowly person at the bottom of the totem pole when it comes to any of this stuff. I'm like, how do I know that? And you don't, it was just, I was shocked.
And
Ginger, you're exactly right. It amazes me when a human doctor, especially one in oncology, doesn't know the term comparative oncology and doesn't. I mean, I think Sean Khanna was the first one to coin that phrase, and it was over 20 years ago. It's been just over 20 years. And I remember the first time I heard it in my young career, I was like, that's just brilliant. And everybody, the immediate thing, and I know you guys tout this, I think it's probably on your website even, about this is not experimenting on dogs and even cats.
Right.
The dogs are the big quintessential model. This is not experimenting. This is allowing me to provide something additional for my patients, another tool that, and fortunately, many times it does end up getting approved and going down that pathway of benefit,
Yeah, fortunately,
I was at with Puppy Up, we do a lot of outreach kind of stuff, and so we were at a craft fair here local to my house, and I was giving people, like, we have little cards for the early warning signs of canine cancer. And the things that I say for comparative oncology is like, dogs get breast cancer, mammary tumors and breast cancer are biologically the same. According to the ladies at Princeton, the doctors at Princeton, I always put that in there so that people don't look at me like that's what you say. And I was saying that to somebody, and this guy looked at me and he goes, wait, what did you say? And I repeated myself, and he goes, I'm an OBGYN. I should know this. I'm like, yes, sir, you should know this. I should not be the one telling you this.
Fortunately, on my first walk, after I lost my first dog to cancer, osteosarc, Malcolm, I started my first walk from Austin to Boston. When I got to the DC area, I met Sean. And he became sort of my mentor and helped me understand comparative oncology and the one cancer, although we weren't using that term back then, almost 16 years ago. Look where it's come down. Speaking of, we need to get Chan on the show. Ginger,
Yes,
We
gotta
we
get.
have not. He'd be great for this.
Well,
Yes.
I've met him so many times, and he helped with one of the puppy up walks. I think it was in Chicago, and he's just always been a mentor and leading the edge and the edge in terms of our thinking. But we now think in bigger terms in terms of comparative biology, right? The wonderful work that Broad is doing in terms of comparative biology and how we can learn not just cancer, different diseases, and how it relates to us within
Yeah.
our ecosystem. So on that end, to that end, it really is fascinating. In the 16 years since I lost my first daughter to cancer and started my journey, we really have learned a whole lot, and we've come a long way. But it's just not enough. And I want to go back to another point from that symposium on comparative oncology in DC. Is that Big Pharma came out and they were here learning about comparative oncology and what was going on in veterinary medicine. And one of the guys from Big Pharma stepped up and just said, "Hey, that's great what you're doing. That's wonderful. Just bring us validated targets. It's all we care about." And it's all thinking, well, okay, who's going to
Yeah.
you going to finance that? Or are you going to help with that? And so we still have a lot of work to do, but yeah, drug companies now are seeing, but it is interesting that you still have to get that orphan orphan drug designation to get that moving along. Okay, so back to Stelfonta. I think probably what I think is the most important question, perhaps for myself and perhaps for other pet parents, is I want to talk about Hudson in his case. So he was I think we pulled off, Ginger, maybe 12 mast cell tumors over a period of, that's probably on the high side, maybe 8, I think.
No, 12 is probably pretty good because he had multiple surgeries and sometimes we took that more than one was removed.
Yeah.
And fortunately, I also met Craig Clifford early in my travels, and he was the one that recommended doing the target analysis, right? The P2, I can never remember the name of it, the kit where you find out
Yep.
the additional diagnostics to determine, is this do we have slow mitotic growth, or is this a faster growing tumor? Fortunately, in Hudson's case, they were all very slow growing tumors, and we were able to excise them, take them off. We got clean margins every single time.
Yep.
So here's, I think, what I think is perhaps the most important question in a case like Hudson, where if you do the responsible thing, Ginger, your dog, your fuzzy butt, develops a lump or a bump, you take him to the vet, it's
Yep.
even if it's just as big as a pea, take him in, get it aspirated. It comes back, it looks like it's a crazy tumor, and they do the additional diagnostics on it, and they find out it's slow growing, and you take the tumor off. So the question I have for you is, as a pet parent, and you do get that dreadful but hopeful diagnosis, mast cell tumors are still, I think, that have some of the better outcomes of the other
Yes.
cancers. But when you do get that diagnosis as a pet parent, when do I just, okay, now's the time to take it off, and if we get clean margins, we don't have to worry about it. We just, every time we see another bump or a lump, we take him in. What is the time that we do that, and when does Stelfonta come into play?
It's a great question. And I will tell you, that's probably the biggest thing about oncology is I tell people no two pet parents, no two pets, no two patients are the same, right? So it's going to be different. And there's a lot of choices that go into it. Now, what I do say is I agree with everybody on cytology. Biopsy was the gold standard for almost everything, especially when I was young. And now, well, you heard about liquid biopsy. We've got newer tools, and one of the first things I said to this company when they reached out to me and said, "Hey, we're looking for an oncologist." I said, "Wow." I said, "Well, there's several things with your drug I want to talk about, but the first one was you're going to be up against no tissue biopsy," which is not entirely true because you can take a tissue biopsy, but you're not going to have margin assessment, right? And that's one of the things we always look at. Now, what I will also say over the last 20 years that I've learned is we used to call narrow margins, those 1-2 millimeter margins, we used to call those bad guys. And we would set out and do 15 to 18 doses of radiation or second surgery. Now we know that the low grade ones, narrow margins may be just enough, because they don't recur as often as we think they do. These are tough questions that we've been asking ourselves for 15, 20 years. And I even say, wow, I was a really good radiation oncologist. I cured a lot of mast cell tumors, but I treated a lot of them that probably didn't need treatment now that we know more, right? And I think that's the key. Is same with biopsy. We call biopsy gold standard, but I'm going to argue with that and say no, because now we use the ancillary tools. We use immunohistochemistry. We're now using genomics. We're taking parts of these tumors and doing genomics. And I heads up, that's what they do for you and I right now already. Every single
Yep.
tumor they take off of, they don't even ask. They go for genomics because they want to know what's happening inside that tumor and whether it does require more aggressive therapy. And I think that's the beauty of us. That's the beauty of us evolving and learning. You're exactly right. The two local treatments I think of, surgery and Stelfonta. That's what I think of now. And when I say no two pet parents are alike, sometimes it's a matter of, hey, I really don't want my dog to undergo another surgery. We just took 6 of them off 6 months ago. Can we just, treat them with something simple versus surgery? I used to say we made our dogs patchwork quilts, which we did. I've sent dogs home with 9, 10 little incisions, just because they had them all removed at once. Maybe the owner doesn't like anesthesia. Maybe the pet doesn't tolerate anesthesia. So those are all reasons to say, maybe we could do Stelfonta. And if we need a light sedative, we can, but we don't always need a sedative. Those are good choices. And again, I always say there's tumor factors, there's patient factors, and there's owner factors that make my decisions. And I should say pet parents, those are the ones that help me make my decisions. And the other thing is, and I actually had an Italian oncologist tell me this, because I said, how do you get around no biopsy? And she said, right now we do cytologic grading. It came up, it's been over 10 years since it came up. We're getting better at understanding mast cells when it comes to cytology, where we look at a bunch of it and we say, yeah, this looks more aggressive or less aggressive. But the other thing she said, when was the last time you made recommendations to an owner based off of one diagnostic test? And I was like, ooh, good point. Never. And she's right, because even if a biopsy tells me this is a low-grade mast cell tumor, it's not going to do anything. I will tell you, if that patient came in with a history of ulceration, bleeding, swelling, those kinds of things, the first thing I'm going to say is, gosh, it's behaving badly. I don't care how good it looks or what kind of a player. It's, it's a wolf in sheep's clothing, basically.
Well, you're right. And I think someone explained it to me perfectly. They said, when they were looking at Hudson, they said he's probably going to get a bunch of these mast cell tumors perhaps over his lifetime, and hopefully they're almost all very low grade. But you're always worried about the unpredictable one. You're always worried about the ticking time bomb one. And so you're recommending, even with like a low grade mast cell tumor, you're recommending doing the in conjunction multimodal treatment with Stelfonta. Is that correct?
Yeah, well, again, it depends, because if it's low grade, you may just need Stelfonta for local tumor control, just like you just need surgery for local tumor control.
Okay, so there are times when you recommend just Stelfonta and not even taking the tumor off. So it's that.
Absolutely.
And so Stelfonta then completely obliterates the tumor. It lyses, I would imagine, and then it heals up nicely.
And this is where it turns veterinarians on their head. It turns owners on their head too at first, but basically that tumor dies and it doesn't just go away. It falls off, sometimes gross, but the dogs sometimes eat it, but and this is what it's supposed to do. So over a period of 3 to 7 days, it dies, what we call necrosis. It turns black and it basically shrinks away, and then it falls off, right? And what's left behind is this wound. Well, we don't go in and surgically close the wound. It's actually truly what we call hands-off healing. You don't help it heal. And that's the hardest thing for vets, because from the time we're, young, we learn, what do you do with wound? You manage it, you clean it, you bandage it, you put stuff on it, you add stuff, so you want to close a wound as fast as possible. This drug has so many healing properties, you don't have to do all that. And the other thing is too, if you look like clinical studies and such, we don't recommend an e-collar. I mean, we just say if they want to lick it, they can. Most dogs don't. After that tumor falls off, they pretty much ignore it. Now, there are incessant lickers, and I don't want to call out Cocker Spaniels and give them a bad name, but some of them are just incessant lickers, you know, and that's when you do have to use a collar because they just mechanically traumatize anything.
What I mean, this is powerful because so there's some times where you're saying you don't even need to take it off surgically. We'll use Stelfonta, and that's important because Hudson had so many over his lifetime that the one that eventually killed him took him out. The initial tumor came back at the very same place. One was taken off at the end of his tail, at the very I'm sorry, the base of his tail. It was the base of his tail. And I had this the surgeon. He was an oncological surgeon. He was a surgical oncologist. He was just a GP surgeon in Memphis. There were only two in Memphis at the time. And I said, just do radical surgery, right? Take it, back in the old days, take out as much as you need to because I want clean margins. Well, he was very aggressive. I mean, very aggressive. He took out a lot. And the problem was the tumor that was a part of a cluster of tumors that ended up killing it grew right back at the same place. And it was very tough for the surgeon to then go back, and they didn't have enough area to remove the tumor. So you would not have that problem with Stelfonta.
Yeah, it's interesting because again, the big thing is Stelfonta getting margins, by how it works chemically, locally, that kind of thing. And yes, it is by its targeting and such, it is getting margins, but the proof we have of that is when we look at how many patients don't have a tumor a year down the road, 2 years down the road, 3 years down the road. So they're essentially cured of it. And I will say, because I've plenty of friends that are surgeons, they will always cure more cancer than any of my medical therapies, anything I do. That's just, they take tumors out and put them in formalin. That's what they do. And, too often the other things we rely on, whether it be chemotherapy or intratumoral therapy like Stelfonta, or we rely on radiation, it just takes one rogue cell and we fail, right? So one little outlier on the edge. I have had tumors like what you described treated with Stelfonta successfully. And I think you mentioned one of the big things, what ended up taking him out. I'm assuming he eventually metastasized.
It did to his spleen, right? Right. It's part of the blame rests upon my shoulders, and I carry a lot of guilt for it, is that when it came back there, I didn't, and the veterinarian we had in Memphis just wasn't educated to know enough that you should do an ultrasound as well. And so we didn't do an ultrasound and catch the mast cell tumor that was in his spleen. And by the time we administered chemo, it was just too far gone. And so it metastasized. We tried to treat it, and he had a severe reaction to the chemotherapy, is what we think. It just basically a complex system failure is what resulted in. He was just not savable. So I've been trying to talk about it with that. I'm still have a very hard time with Hudson because some mistakes were made, and I carry the blame for a lot of that. But education is so important with what you're doing. But before we get into the importance of educating veterinarians, GPs, we know there are a lot of them out there to educate, but they need to know that we have Stelfonta and the quiver of arrows, therapeutic arrows that we have out there. Before we get into talking about educating veterinarians and even veterinary oncologists, let's talk numbers. What are your success rates and what are your outcomes with Stelfonta?
Yeah, so with one intratumoral injection, 75% complete response rate. And if you do two, that increases to 88%. So those that don't respond fully to the first one, if you do a second injection, that brings that response rate up to 88%. Now, that's not the 90-something that surgery touts, right? Again, surgery is always going to cure more. However, if you compare it to other things, and I don't want to pick on my friend Palladia because I like Palladia. And you talk about clinical trials. I was at a practice. We did the clinical trials when it was a number. So I know the response rates. But if you really look at Palladia response rates, their complete response rate is 8%. And if you add in partial response, it brings it to like 37%. So and I'm talking a complete response of 75%. And the only thing that speaks above that would be surgery. It's the only thing.
Hmm.
Now, I don't want to put this into the category of, it's not for every tumor. Absolutely not. There is an indication. It definitely does not treat anything metastatic. In fact, it's labeled for non-metastatic mast cell tumors. Grade doesn't matter. But it is limited to cutaneous anywhere on the body. And when you talk about subcutaneous, that's below the elbow or hock. And part of that is the way the drug works. We don't want dead tissue trapped underneath the fat tissue and such on the body. But the important thing is educating veterinarians when it's appropriate, and as well as, because the target is really primary care, general practitioners to use this drug. I have oncology friends that use it all the time. However, they're using it on tougher tumors. They're using it sometimes off label because they know the dog has lymph node metastasis or something, but they're combining it with chemotherapy, and they're combining it with other therapies to treat the systemic aspect. This is again, I think if it fits the label indication, it's an appropriate drug to consider. And it's an appropriate drug to say to the client, hey, I can do surgery or I can do this. And some things I used to do surgery on some things, and some things I did radiation on. Sometimes it's going to be one or the other is going to be better, right? It's not going to be, you can do either or, it's going to be one or the other.
Right. Well, I was amazed by your success rate when I went to your website. So I think that's a very high number. And you're right. It's just not as high as surgically excising and taking the tumor off. It's not that high of it. But as a pet parent, when I'm posed with that question, should I do a surgery or administer Stelfonta? One of the questions I'm going to have is, now if we try Stelfonta and it isn't successful, are you seeing any shrinkage of the tumor or the mass, or is it responsive at all, so that it's not going to do the exact opposite and grow, and now I've got a bigger problem, but it at least keeps it, either contains or shrinks it, so that it's less of a mass for the surgeon then to go and take it off. So is there any shrinkage? Are you seeing any reduction or at least abatement of growth?
The answer is yes and no. So yes, it can shrink them down. And in fact, I've had oncologists tell me, oh, I used it to shrink it down to where the surgeon could do surgery. As well, if you have a resistant or an aggressive mast cell tumor, they can grow in the face of anything you give to them, right? So, and that's the thing that we don't want to do, is we don't want to stimulate it to grow, right? We don't want to do ever anything that will stimulate it to grow. And there's no evidence out there that Stelfonta would ever do that, so I think that's the key. And I have had, again, the clients say, well, gosh, as a pet parent, which one's better? And I'm like, well, you just have to be educated. You have to look at all the pluses and minuses. The wound, you would think, and I had veterinarians telling me this, oh, my client will never put up with that wound. And the first thing I say is, really, because I did radiation and caused a lot of burns in dogs over the years. And as long as I educated them, and back in the day, we pulled out a photo album with little pictures and showed them radiation side effects, wherever their dog was going to have and what it would look like. And the same thing with Stelfonta, you have to do that. Unfortunately, that's why we have websites, education websites. I mean, I told you, I said a lot of things to them when they asked me. And number one thing I said, this is education. And you got to educate veterinarians, you got to educate pet parents, because it's such a different drug. It's so different than anything we have. And I've put a lot of chemotherapy intratumorally into tumors of the mouth, soft tissue sarcomas, you name it. And most of the time it causes a sore. I mean, really, it's pretty worthless, even though we tried it because we had nothing left. This was the first time besides radiation where I actually put something in a tumor and I was like, whoa, now we have no tumor. This is great. But now I have a wound. And the good news is the wound heals, right? And social media is telling me otherwise when it comes to parents, pet parents, and how they feel about the wound. I'm shocked at how many of them are like, it worked. Look at my dog, he's healing so fast. And, and I think that's one of the big things that shocked me. I mean, when I first started getting consults of these wounds and things, I would literally, I remember my eyes going, whoa, I'm not so sure about this one, but I relied on one of our senior vets in Australia who's treated over 800, and Justine be like, no, that looks great. That's day 5. That looks excellent. And now I'm the one that someone sends me a picture and I go, oh, that's so amazing. That's beautiful. Look at that healing. And yeah, but it's a lot of education, because I had an oncologist the other day call me and say, Pam, my surgeon wants to go in and do skin grafts and this and that and the other. And I'm like, do not let them get near that wound. Give it a few days. And sure enough, a few days later, she's calling me saying, yeah, I can't believe how much it's healed in 3 days. I'm like, I know, you just have to be patient.
Yeah, the point I wanted to get across to the audience is that I don't want you to look at it as either or, right? You're either going to do this one, just try, perhaps maybe if this is recommended, try the Stelfonta first, and then if that's not responsive, then you have the option of surgery then after that. So it's not a binary choice at all.
Absolutely, absolutely, it's not. It's it's just, if it works, great. If it doesn't, you can say, well, hey, let's jump in and get surgery done before it gets bigger.
Absolutely. How long has Stelfonta been on the market?
So we are over 3 years in the United States now, and nearly 4 years in Europe.
Wow. So you've been on the market 3 years in the United States.
Yes.
You would have been a therapeutic option for Hudson. That's interesting. So that go back to education. I don't think we heard that at all from any of our the veterinary oncologists with which we're friendly. But again, they're busy educating the GPs.
Yeah.
Well,
Go ahead.
And
My question-
veterinary oncologists, and you mentioned Craig, and Craig was on our scientific advisory board, and so was Phil Bergman, and and again, it's so novel that it's been a very careful education of veterinarians, and it is probably, I shouldn't say this out loud, but it's probably the first drug on the market where myself included, I wouldn't go off label until I was really comfortable with it, because you need to follow the label instructions. You need to follow everything on there to make sure that you have a good outcome, and it really is a case selection, owner selection, that kind of thing. And that's not to say it's complicated. It's just, you have to give them concomitant medications. And that means Benadryl, Pepcid, things to protect them from mast cell degranulation, and you have to start that before you give the drug, not after. And you can't skip doses. If the dog doesn't really want to eat because they're pain meds or because they just don't feel well that day, you got to get those medications in them.
That was the word I was looking for, degranulation. That Hudson had this massive degranulation and it went wild. So I a part of me still tries to block out that part of my memory.
Hey, and you say you carry guilt with you. I'll tell you what I tell lots of people, because I've dealt with, not only my clients, but also my own pets. You did the best you could with the information you had. And that's where I think most pet parents understand, because that's the key, is education. It is. I mean, that's what oncologists, that's what we do, is educate. That's primarily our job.
Well, I think that it's been part of what inspired Fuzzybutts and Friends, is that we just need to have more oncologists talking about the newest. We have Phil on every year to talk about what's going on, what's excited, what are your Gil Vatnib was his, he was on, I guess, at the end of last year talking about that. Very excited. So and I tell him, anytime you have something new that's coming out, maybe it's not in the market for 6 months, come on the show and let's talk about it, because that's our job, is to educate. We cannot, education and awareness has to be a fundamental component of everything that we do, because getting information out is tough. Ginger, you had something a second ago. I didn't want to interrupt you.
Yeah, I did. So what I was asking is, I know that I'm sure you market, obviously, to the veterinary oncologist. Do you ever market to the pet owner?
That's a really good question. And, marketing to the pet owner is allowed in the United States. We have not launched any campaigns directed at pet owners. However, Dr. Sue, cancer vet, she's one of our leading speakers and such, Verbac, who is our partner that distributes the drug. They contract her to do speaking and sponsor her. And she touts all of her social media, trying to educate people and pet parents. As well, she has her own couple of pages on Facebook, specifically one for veterinarians and one for Stelfonta for mast cell tumors, and that's for pet parents, not for veterinarians. And so it's almost, I call it the little support group for Stelfonta, because the parents are really educated and they educate each other. I'm sorry, go ahead.
I think that that the pet parents are missed in a lot of this. And because I know that even, Dr. Sue, she's not hitting everybody, because if your dog's never had a mast cell tumor before, you're not looking,
That's right.
So, and even when the Pet DX and the Onco Canine test came out, I knew about it because we knew about it. And I went to my veterinarian and I was like, hey, can we do this? And she's like, what are you talking about?
Yeah.
Because she's a GP, and she was like, well, my rep, my drug rep was just here yesterday.
Didn't
Why
bring it
didn't
up.
he tell me? And he didn't
Yeah.
bring it up. And so, and we just did a podcast with a really nice lady that's with the Dog Scouts.
Mhm.
And so she's really promoting that. So
Mhm.
if there's a way that we can help promote it and get it out there, we have our walks, we have our newsletters, we have our website. Because that's what I really, that's part of our education that's in our mission. And I
Yes.
feel that somehow we're missing that connection, because the pet parents, like you said, they'll deal with, I mean, they dealt with the radiation because they knew what that was a chance to give their dog life,
Yep.
a better life, whether it was 6 months or 6 years or whatever it might be. And it's just somehow there's a disconnect. I even asked somebody that's coming out with a new diet, for dogs with cancer, and it's a liquid diet. It has to be prescribed by your veterinarian. But if all the veterinarians don't know about it, they don't know to prescribe it. And if the pet parent doesn't know to ask about it, there's a 6 of one, half dozen, there's a whole disconnect there. And somehow or another, I want to bring those together.
Well,
I agree.
whatever
And
I
I
can
think it's a
do.
great mission, because it's something that, veterinarians every day, I think you said it, Luke, look how busy oncologists are, and they're trying to stay up on things, and same thing with veterinarians. And I agree. I mean, one of the sites QBiotics itself, not Verbac. Verbac has their site for Stelfonta, but we did a site called Stelfonta.com, and it's directed at pet parents so they can educate themselves on mast cell tumors, on everything, and it's got pictures, and it's got even testimonials from previous owners that have used it for their dogs, and journeys, literally journeys of the photos and the pet owners and how they felt throughout it. And even educational, there's things to download to keep track of your medications, and but that's probably the most marketing we've done, towards pet parents, except for breeds. Verbac came to me and we talked a lot about it, and it was brilliant, is to say, can we do newsletters for certain breeds, and it's great, because the ones that need to know are the pug owners and the Boston owners, and quite frankly, all pet owners. But
Well,
pet parents are very resourceful to find information, but we need to also make it available.
we'll count us in as.
You can. I'm sorry. Go ahead.
Yeah, I think you and I were about to say the same thing.
Okay.
Count us in as part of your coterie of advocates for Stelfonta. Whatever we can do, Ginger, I think that's what you're going to say, the Puppy Head Foundation, and we can do here to get the word out, because that's what I look at things as having, as a pet parent, I want as giant of a basket of therapeutic and diagnostic options that I can, and educated people to inform me, we'll do this first. Here's the plan. Help me put together the plan.
I was going to be a little bit more targeted than that. I'm going to be a little bit more aggressive. I'm not going to say anything we can do. What I want is some type of article or permission to steal stuff from your website to put on our website for it, and to put in our newsletter.
And we'll send
And
people
if
to
you have
Stelfonta.
photos, that's what, Luke?
Make sure we send people to Stelfonta.com to learn about mast cell tumors.
Correct.
Yes.
Yeah. Well, that if I have the literature, then it's going to go.
Yeah.
It'll go to the right place, and they can take it to their vet. So yeah, we have a lumps and bumps check sheet that we have everybody. So it has the medical dogs laying out like this, and on the side.
Yes.
And then on the flip side, we tell them how to mark it. Like if you have a black dog, use whiteout. If you have a white dog, use a Sharpie, that whole thing. So yeah, whatever we can do, send me whatever, and we will put it out there.
Okay. Yeah,
Yeah,
that's great. I'll talk to the gal Haley that works with all of our materials and stuff. So
Okay.
yeah. And
Yeah,
I love the lumps and bumps thing because that's like literally it's one of my favorite things to lecture to pet parents about is, you know what you can do? You can be active at home. You can do your part. You can find these things and mark them, and don't walk into the vet and say, I think it was the left side. Do you remember? I can't feel it now. And then you spend 20 minutes and you can't feel it. And, so mark it down. I love it.
One of my dogs was going in for, I can't remember what surgery she was going in for, but I did, like the night before, I got out that sheet and I marked it on the sheet. And then I even wrote on the sheet, marked with whiteout. And then there was another place that says marked with black, Sharpie. And I don't know, I probably found like 8 or 9 of them. And so this is all drawn out, and my vet did the surgery, and he said, oh, thank God you gave me that and you had it all marked, because I would have never found them. And he says, and I believe whiteout is a cure for lumps and bumps. He said, because I could not, he said, you put that whiteout, this was Dr. Blackburn. He said, you put the whiteout like right on top of it. So he said, that really wasn't good, because then I got the whiteout. He goes, but on a couple of them, I couldn't even find the lump anymore. It was gone.
Maybe there's a small molecule in whiteout that you can.
That's what I was going to say. Maybe I didn't investigate that.
Investigate that and then let me know.
Maybe that's why all my cancers with my Great Pyrenees have been terminal, because you can't use whiteout with Great Pyrenees because you won't be able to see the actual spot.
Oh no, I used red Sharpie on those.
That's right.
Because the red they're going to think they're bleeding or something, so you're not going to miss the red. So
I think on one of the early ones you did use mascara on Hudson, though.
Oh, I did. Yeah, I did. I used
mascara.
Yeah. Yeah. I typically saying on here that lumps and bumps is the hill I'm going to die on, because I'm constantly, I'll pet somebody's dog and I'll say, did you realize, because it's like my hands find them, right? It could be a Great Dane, a Great Pyrenees. As soon as I put my hands on it, it finds the lump. And I'll say, oh, did you know, I'm sure your dog has this lump. And then sometimes they'll say, yeah, they've had it checked by their veterinarian. And then sometimes they say, well, yeah, I took him to my vet and they said to watch it. And I'm like, so did they do a needle aspirate of it? No, they just told me to watch it and let them know if it changes.
Yeah.
What are we
watching
watching
it turn into
for?
cancer?
Watch it turn into cancer, and then
I
then you got a bigger mess and more expensive mess. Yeah. Well,
yeah, I'm just like, no, we're not watching. I'm like, no. I try to be really diplomatic, because here in Memphis, I know most of the veterinarians, but I'll say, oh, who's your veterinarian? And there's 1 or 2 that pops up. I'm like, when you go back, tell them that Ginger, remember my name, red hair, Ginger, really would like for them to do a needle aspirate of that.
Well, maybe it's a branding issue you have, Ginger. Maybe instead of your brand being your little dog, maybe it should be the queen of lumps and bumps. If you're the queen of lumps and bumps,
I like that.
Everybody will know you and remember you.
That's right.
Well,
the queen of lumps and bumps.
There's so many things that I wanted to get to. We try to keep this at an hour, and I'm so fascinated. I have so many other questions, in particular about clinical trials. But we'd love to have you on the podcast again, especially as you continue to conduct more studies and do more research and see it in practice and some of the outcomes with us. But I just couldn't help but have a question, and that's why I asked how long Stelfonta had been on the market for 3 years. I was curious to see if you were learning that you were having any recurring tumors reoccur on the original tumor site, which can occur with surgery, and that's exactly what happened with Hudson. So, but you guys your company might not know that for a long time, but it's interesting.
Yeah. The only thing we can base it on at this point is because it's one of those things that it would take, 1,000, 2,000 dog trial, and you'd have to follow them for 5, 6 years. And it's just so cumbersome. But based on one of the things that I think is very important and most veterinarians aren't so aware of is something we call pharmacovigilance. And that is where if you see something unexpected, or something abnormal with a drug, and it doesn't matter whether it's a vaccine, it's a drug like Stelfonta, or it's an antibiotic, you should call the 800 number and report it, right? And nothing says it more than COVID vaccines when we were having reactions to those and such. And what that does is that provides more information as the drug is on the market. And one thing it does, it either supports, hey, the drug's doing exactly as we saw in studies, right? It's safe, it's efficacious, nothing's changed. It will also tell a drug company, oh, wait a minute. And a really old example is Rimadyl. When Rimadyl first came on the market, I was a baby veterinary technician. And all of a sudden it was like, wait a minute, does it cause liver problems in higher amounts than we thought? And lo and behold, yes, in certain breeds and certain color, but it's not a bad drug, but that's something they learned. And one of the things that pharmacovigilance, believe it or not, is lack of efficacy. And that fits in that as if veterinarians call us and say, hey, I treated with Stelfonta 2 months ago or 6 months ago, it came back. And a lot of times they're just asking, can I retreat it? We mark that down and say, okay, we need to follow those. And it's not any higher than what we see in, surgery, what we've seen for the last 20, 30 years with surgery. It's probably lower than radiation, because radiation, we're treating such a large field usually to kill everything. But yeah, we are seeing recurrences, but again, it's not any higher than the usual, what we would expect. Yeah.
Are you, in that vein, are you seeing any side effects of Stelfonta that are being reported, or any problems out there that the pet parents should be aware of?
Yeah, so one of the things that, again, probably the largest number of pharmacovigilance reports we get is when there's excessive swelling, of the site. And again, mast cell tumors, you can look at them funny, you can stick a needle in them, you can do a lot of things, and they could degranulate. And
Get angry. I call it getting angry.
Yeah, they get angry. They
get angry.
do. And now we're putting a drug in it that's going to cause destruction, right? So if you think they're not going to get angry at that, they are. And that is one of the biggest things that we get calls for. And, I would say probably more than 75, 80% of those calls are because someone didn't do the concomitant medication. So they didn't do the low dose of steroids for a few days. They skipped H1 or H2 blockers. They just said, they're not that important. No, it's really important. And again, one of the things I think about, we've learned, like if a vet calls me about a tumor on the lower part of the limb and it's subcutaneous, I'll say, yeah, why don't you increase the H1 to 3 times a day? And this isn't on the label, but these are things we've learned that can help mitigate that risk. Keep the dog. Pain is a big thing that I tell people. Veterinarians, be preemptive in your pain. Treat it just like you would if you were doing a surgery. Don't say, oh, it's no big deal. It is a big deal. I mean, we're causing a wound. And that keeps the dog up and mobile and moving and doing their normal things. Yeah.
Well, it's certainly not nearly as severe as some of the adverse events of some of the newer, more promising drugs that are out there. But it's just a risk-reward assessment that you have to make with every treatment option. You have to make a treatment option with radiation. I had a nasal adenocarcinoma dog as well. My second dog that got cancer, and it was so burning the roof of the mouth. That was all the side effects. That was just it's part of it. So as we wrap this up, Dr. Pamela Jones, thank you so much for coming on the show. But is there anything else that you would like to leave our audience with? Something you'd like to let them know?
Yeah, it kind of goes along the education. Don't be afraid to go research things on your pet. And I'm going to say there's a lot of misinformation on the internet, but there's a lot of good information. You got to go to reputable sites and ask your veterinarian. I had to train my mother to ask her own doctor questions, because she's like, well, he gave it to me and he wears a white coat, so shouldn't I just believe him? I'm like, no, you need to have a relationship with your veterinarian. You need to have a relationship with your doctor and ask those questions. Don't suffer in silence,
And I definitely go see a board certified veterinary oncologist, because we know a lot of them out there. Of that 400 plus, we know a lot of those, and they're very relationship orientated. They truly do. The vast majority of them, there are a few exceptions to that. They truly develop, I think, a long-term rapport with their patients, and that's such an important feedback loop, but also an educational loop as well. And then go to Stelfonta.com. Is that the website?
Stelfonta, Stelfonta. I like how you want to put a B on there.
I knew it. I grew up listening to Harry Belafonte.
That's what I was going to say.
Well, my mom was a big fan of his, and I knew prior to this podcast I was going to make that mistake, and I sure enough,
it's
I
great.
screw up. Yes.
I thought you said that earlier, and I was like,
Yeah.
Did he say that with a B?
I'm dyslexic amongst other pathologies. My apologies. But so go to Stelfonta.com. That would be certainly the starting place to learn more about it and share that with your GP. But the first step is your GP to get it aspirated. Whenever you have a lump or bump, even if it's the size of a pea, that's the time to take a look at it. All right, Ginger. That we're wrapping up this episode. So is there anything else that I screw up anything, or aside from the name of the drug?
You didn't call her little dog, so
Exactly.
That's
That's
right.
queen of the lumps and bumps. But
Little lumps and bumps.
but
I like
I
it.
have this, like I have a visual of me in like a Michelin man, lumpy bump kind of thing.
Hey, I'll make a logo for you. I'm an artist. I'm a legit artist. I will totally do a logo for
I can see it now.
there you go. I enjoyed chatting with you too, and I'd be happy to come back, anytime and update you on things as well. Yes.
Absolutely, Dr. Jones, Pamela, and also more importantly, if there's any more small molecule research that you're doing on the veterinary oncology side, we'd love to have you. Is that where you're going to primarily stick in veterinary medicine in your pipeline, or oncology drugs, therapeutics? And
No, actually, we have a wide therapeutic pipeline. And the next one is the wound healing drug I mentioned. We actually have some anti-infectives and anti-inflammatory. So it's kind of targeting a lot of different things. I think oncology just happened to be the first one that went down that line. But yeah, a wide variety.
or is that on the veterinary medicine side or human medicine?
Both, actually.
Well, we'd love to have you back on the show to talk about that. I mean, our foundation is focused, since I lost my dog to cancer, and that's how we started. We focus on comparative oncology, but on podcasts, it's all animals, all fuzzy butts, and all issues. So we would love to have you back on the show and talk about QBiotics' other work in veterinary medicine. We'd love to have you on for that.
That sounds great. Yeah,
Excellent.
I'd love to.
Excellent. All right, everybody. Thank you. And obviously, we didn't have to do a cancer tip of the week, sponsored by the Puppy Up Foundation, because this entire episode was a cancer tip of the week.
Was a cancer tip.
From the queen of lumps and bumps. All right, everybody. Thank you so much for joining this episode of Fuzzybutts and Friends. You can watch us on our YouTube channel. It's fuzzybuttsstudios.com, or check us out on your podcast platforms such as iHeartRadio or Spotify. Man, that's all alliterative. Sometimes it just jams me up. Thank you, everybody.
All right. And you need to talk
Thanks
about Jeannie's Therapeutics.
Oh, yes. Thank you. I have that on my, I have so many notes. I have so many notes on this. I left so many things I wanted to talk about because I can't even read my notes. So thank you very much for sponsoring to Jeannie's Therapeutics for sponsoring this episode. My Fuzzy Butt, Indiana, takes 2 doses of their wonderful CBD hemp oil formulation for his inflammation and all of his problems as a geriatric dog. So I highly recommend you learn more about Jeannie's Therapeutics at geniestherapeutics.com. Until next time, everyone, puppy up. Talk soon. Bye-bye, guys. Thank you.